In the early 1900s, heredity still felt like a black box. Scientists knew traits passed from parent to child, but they did not know what carried the instructions. Inside cell nuclei, researchers found a strange substance first called nuclein. It held phosphorus and behaved differently from familiar proteins, but it looked dull and repetitive. To most biologists, it seemed far too plain to hold life's secrets. Proteins, on the other hand, looked like perfect candidates. They were built from many different amino acids and could fold into countless shapes. That variety suggested complexity, and complexity sounded like information. So the assumption hardened into a working belief. Proteins carried heredity, and DNA sat nearby as support material. Many experiments followed that bias. DNA often became background, measured but not truly suspected. That early judgment shaped the next decades. If DNA was simple, then studying it promised little reward, and the most ambitious minds aimed elsewhere. Genetics grew as a science without a clear chemical identity for genes. Meanwhile, DNA stayed labeled as a basic nuclear ingredient, not the master plan. It was a quiet mistake, because the search for heredity's mechanism started with the wrong lead. In 1928, a British doctor named Frederick Griffith tried to understand a killer, the bacterium that caused pneumonia. He worked with two forms of the same microbe. One form looked smooth and often proved deadly. The other looked rough and usually stayed harmless. Griffith expected the difference to stay put. He assumed dead bacteria stayed dead. He tested that assumption step by step. The rough, harmless bacteria stayed harmless when injected alone. The smooth, deadly type killed as expected. Then Griffith heated the deadly bacteria until they died, and that no longer caused disease. But when he mixed the heat-killed deadly bacteria with the living, harmless ones, the result shocked him. The harmless strain turned lethal. Something from the dead bacteria had survived the heat and changed the living ones. Griffith called it a transforming principle because it transformed one bacterial type into another, and the change lasted. It passed on as the bacteria multiplied, as if a set of instructions had moved between cells. Heredity, it seemed, could travel as a chemical substance. The question became, what chemical? By 1944, the mystery from Griffith's bacteria still hung in the air. Something had moved between cells and permanently changed what they became. At Rockefeller Institute, a physician named Oswald Avery took that problem personally, and he worked with Colin MacLeod and Macklin McCarty. They broke the deadly bacteria apart and chased the transforming substance through one careful purification step after another. The key test sounded simple. If they destroyed one kind of molecule, did transformation stop? They used enzymes that chewed up proteins, and the bacteria still transformed. They removed fats, and it still happened. They attacked RNA, and the effect remained. Then they used an enzyme that specifically cut DNA. This time, the transformation collapsed. The ability to pass a trait tracked with DNA, not protein. That result did not just solve Griffith's puzzle, it forced a shift in what genes could be. DNA, once dismissed as too plain, suddenly carried the power to store instructions and remake a cell's behavior in a stable way. Many scientists hesitated, wondering about impurities or exceptions, but the logic kept pointing in one direction. Heredity had a chemical candidate, and it looked like DNA. By 1950, DNA no longer looked like a lab curiosity. It looked like the carrier of heredity. But one problem stayed stubbornly in the way. Nobody knew how that chemical was built. Into that uncertainty stepped Erwin Chargaff, a biochemist who did something almost unfashionably direct. He measured. He took DNA from different organisms and carefully counted its chemical parts, one sample at a time. DNA was made from 4 building blocks, known as bases. Chargaff compared how much of each base appeared in each species. The numbers shifted from organism to organism, so DNA was not a monotonous repeat. Then his key pattern appeared. The amount of one base, adenine, matched the amount of thymine, and the amount of guanine matched the amount of cytosine. It kept happening. It stopped looking like chance. Those simple equalities changed the hunt. Any structure for DNA now had to respect them every time in every species. It was the first hard set of rules, not a guess and not a rumour. The bases did not float in any proportions they pleased. They came in linked partners. Chargaff did not hand over a full shape for DNA, but he handed the field a locked door with a specific keyhole. In 1951, the problem was no longer whether DNA mattered. The problem was shape. To solve it, scientists turned to X-ray diffraction, a method that treated molecules like hidden objects and used scattered X-rays to map their outlines. Maurice Wilkins worked on DNA fibers that could be aimed at an X-ray beam. The patterns that came back were not pictures, but they carried a message. Rosalind Franklin arrived and pushed the technique harder, treating the fibers carefully and measuring the results with strict precision. When X-rays passed through aligned DNA, the spots on the film formed crisp, repeated features instead of a blur. That clarity mattered. A fuzzy pattern allowed many guesses. A sharp one ruled guesses out. Franklin's data showed regular spacing along the molecule and a strong cross-like signal that pointed to a helix. Wilkins also produced diffraction results that supported the same direction. Now the evidence did not just suggest order, it demanded it. The patterns implied DNA was not a loose chain or a flat sheet, but a structure that wrapped around in a consistent spiral with repeating distances that could be measured. For anyone trying to build a correct model, this changed the game. Any structure had to match those numbers, or it failed. Early in 1953, a new jolt hit the DNA race. Linus Pauling, already famous for solving tough molecular structures, announced he had a model for DNA too. People paid attention immediately because Pauling's past work usually meant he was right. For everyone else, that was pressure and a threat. If Pauling had the structure first, the puzzle would be over. But Pauling's proposal did not match what DNA seemed to demand. The chemistry did not line up cleanly, and the model clashed with the hard limits coming from X-ray diffraction. It was the kind of mistake that mattered because it showed how easy it was to force a neat-looking idea onto the wrong evidence. A wrong answer from the best scientist raised the stakes for everyone. Now the competition sharpened into a narrower question. What shape could satisfy everything at once? Any believable structure had to respect how DNA's chemical parts carried electrical charges, how they could stack and bond, and what the X-ray patterns allowed. Guesswork no longer looked brave, it looked dangerous. The moment demanded a model that fit the data, not a model that merely sounded plausible. By 1953, the pressure built. The chemistry had to add up, and the X-ray patterns had to make sense. Two researchers, James Watson and Francis Crick, tried a different approach. They treated DNA like a puzzle you could assemble piece by piece until every constraint fit. They stopped guessing wildly and started matching. Every wrong arrangement created a clash, a gap, or a shape that simply could not exist. They focused on the bases, the letter-like parts of DNA. Some pairs seemed to fit cleanly, like matching shapes. Adenine lined up with thymine, cytosine lined up with guanine. Any other pairing distorted the whole structure. This was the breakthrough. Two long strands could run side by side, and the bases could link across the middle in fixed complementary pairs. Order could vary, but the pairing rules stayed firm. From there, the double helix fell into place. The paired bases sat inside, protected, while the backbone formed the outside rails. The twist explained the helical evidence, and the constant width came naturally from the specific pairs. DNA no longer looked like a boring chemical chain. It looked like a system, a structure that could hold information in the sequence of bases and keep that information organized with strict pairing. In 1953, the double helix did more than explain how DNA looked. The follow-up papers spelled out what the pairing meant. Each base only matched one partner, by shape and by chemical fit. That simple rule turned the structure into logic. If the two strands ever separated, the code did not vanish. It stayed exposed, letter by letter, ready to be read back into a new strand. That idea changed the problem overnight. Heredity no longer needed a mysterious copying force. It needed separation and matching. One strand could act as a template because it already carried the instructions for rebuilding its partner. Wherever there was an A, a T had to go. Wherever there was a C, a G had to go. Copying became a consequence of pairing, not an extra invention. In 1953 and into 1954, the team pushed this implication hard because it connected structure to biology in a direct, testable way. A double helix did not just store information. It carried its own method for making a second version. Each original strand could guide the construction of a new mate, producing two double helices. The elegance was also a claim. The secret of replication had been hiding in the bonds. By the late 1950s, the double helix suggested a simple promise. Split the two strands, and each one guided the rebuilding of its partner. But it still needed proof. Three outcomes stayed possible. DNA could copy by keeping the old helix intact and making a new one beside it. Or it could break and mix old and new in every strand. Or it could split, half old and half new. In 1958, two researchers, Matthew Meselson and Franklin Stoll, found a way to track old DNA and new DNA without changing what DNA did. They grew bacteria in a heavier form of nitrogen, so the DNA became dense. Then they switched the bacteria to normal nitrogen and let the cells copy their DNA. After each round, they separated DNA by density. After one copy, the DNA did not stay all heavy, and it did not become a scatter of many densities. It formed one clear middle band. Exactly what happened if each new double helix carried one old strand and one new strand. After two copies, a light band appeared alongside the middle one, as predicted. Semi-conservative replication moved from a tidy idea to a tested mechanism. The double helix had not just explained heredity, it had handed biology a working rule. Information lived in a sequence, and copying followed pairing. Over time, that rule became a tool. Scientists read DNA by turning its chemical letters into data. One stretch at a time, then faster, then at massive scale. Genomics emerged from that momentum. Whole genomes got compared, and hidden links between traits and genes became trackable. That same logic also reached the courtroom. Because DNA sequences varied between people, tiny samples stopped being meaningless smudges and started becoming identifiers. Investigators matched biological traces to individuals, cleared suspects, and connected crimes that had seemed unrelated. The power came from the helix's predictability. If the sequence could be copied reliably, it could be measured reliably. Forensics turned DNA from an abstract molecule into evidence with consequences. Then the direction reversed. Instead of only reading DNA, researchers altered it. Gene editing built on pairing rules again. A designed piece of genetic material found its matching sequence, and cellular repair replaced or disrupted what had been there. That approach made targeted changes possible in plants, animals, and human cells, with real promise and real risk. It marked the farthest turn from the old assumption that DNA was too simple to matter